Hi everybody. This is the first video in Unit 1 of Microbiology at Lane Community College. Before we get into it, I just want to set the scene a little bit. This is me. Hi, I'm recording in a room in where I live, and I do a lot of recording in my office on campus also. But this is just what you can imagine, me in a boring room, thinking about you and what you're learning and what you should be thinking, because that is what I think about all the time. So that's where we're at. Okay, so what I think we should do before we get into the Central Dogma is talk about why we're doing this. Why are we going to be learning about proteins? Why are we going to be learning about transcription and things like that? And it comes down to making you able to make decisions and have power. If you understand the central dogma of molecular biology, you will be able to understand how antibiotics work. You will be able to understand why antibiotic resistance is so hard to fight and why it is such a big problem. You'll be able to understand what viruses are and how we try to stop them, because you'll understand the immune system better, and you'll understand disinfection technology and how it works and what its limitations are. All of those things require you to understand what proteins are, and in many cases how they are made by a cell. And if you look at a diagram of a human cell, once you learn what all the parts are, you start to see the whole cell is organized around making proteins. That is what the central dogma is. So with that out of the way, let's get into it. And before that, we actually need to show you some words. These are words I will say all the time. I will talk about bacteria a lot. Bacteria is a plural word. It is the plural of bacterium. Bacterium is one bacterial cell or one bacterial species, and bacteria is plural of that. So be careful with this, just like you're careful with anatomical terminology. And then another thing I'm going to say, microorganism is more general. If I'm talking about bacteria or fungi or protists or other things, I would say microorganism, but that takes too long, so I say microbe. And the adjective there is microbial. So those aren't really words I'm going to test you on. Those are words you need to understand that I'm going to say. And you definitely need to start practicing bacteria is plural, bacterium is singular. And while we're on the topic of words that I will say but not test you on right now, there are a few really important ones that come up maybe later in the term or maybe earlier in the term. I don't know, but they will come up, and we won't look at them in detail until Unit 3. So I really want to introduce them now. One is pathogen. Pathogen is a microbe that can cause a disease, and they range from highly virulent pathogens all the way down to opportunistic, weakly virulent pathogens. And that virulent part is from virulence, and that is the ability to cause disease. So a highly virulent pathogen can cause disease easily, and it can cause severe disease. So an example would be Bacillus anthracis, which causes anthrax. If you inhale a few of those, you have a very good chance of being dead within 3 days. And the virus that causes rabies, if that gets introduced into your tissues, if you don't treat it, you have a 100% chance of dying. Those are highly virulent pathogens. You could compare those to something like the cytomegalovirus, which you may be familiar with. You may have been infected by it. Most people get infected without any signs or symptoms. So that's a weakly virulent pathogen. Okay, so hopefully those are straightforward enough. And then the important fact I want you to know is where microbes come from. Now, you probably remember using hand sanitizer, and you probably remember wiping down doorknobs and surfaces at the beginning of the COVID pandemic. Because we knew you can get some viruses by touching a surface someone else has touched. And that's particularly true for the common cold and for influenza. Not so much for COVID, it turns out, but definitely for the common cold and for influenza, touching a surface is how you get exposed most easily. And so we think about surfaces, but in the microbiology lab, when you take this class in lab, you're going to be trying to grow microorganisms, and you don't want anything from the environment to contaminate your microorganisms. And what you'll find is that dust is the worst thing. Dust is where microorganisms come from. In little invisible particles of dust will have a fungus on it, or it'll have a bacterial endospore on it. Dust can bring microorganisms anywhere. You can find microorganisms in the stratosphere if dust has been lifted that high. So dust is the enemy, so to speak. Be aware that falling dust brings microbes with it. So anytime you have anything that's open to air and dust can fall into it, it will quickly be exposed to microorganisms. Okay, that's important stuff, but we still haven't really gotten into the big topic. And the big topic is the central dogma of molecular biology. So now that we have finished all of these words you're going to hear, we can jump into the central dogma of molecular biology. Here we go. I end up saying that phrase, central dogma of molecular biology, a lot, even though it is dumb. It is not a phrase anyone should use, but the idea it represents is very important. So here's what it says. All the information in a cell that encodes what the cell is going to be, but mostly what proteins it makes, all that information is stored in genes in a DNA molecule. Okay, that's the first part of the central dogma of molecular biology, as we say all the information is stored in DNA. And if a cell wants to make a protein, it needs to decode that information. And what it has to do is get that information to the little machine that decodes it. And so it makes a small, much shorter copy of a gene. So the DNA contains all the genes, but a messenger RNA will be made that is almost identical to DNA, but it's short. It only contains information for making one protein, usually. So that's a messenger RNA. And making messenger RNA is a process of transcription, and it's carried out by RNA polymerase. So if you're with me so far, that is a lot of the central dogma. The last part is that messenger RNA will be read, so to speak, by a ribosome. And the ribosome pulls the messenger RNA through it and uses a very clever system that we won't get into to figure out which amino acid should go in the amino acid chain of a protein. Because a protein is just a chain of amino acids, but they have to be in a very specific order. And that order is what's determined by the DNA. So the central dogma of molecular biology is DNA stores the information for making proteins, and transcription makes a copy of those instructions in the form of messenger RNA. Translation makes the protein based on what's found in that messenger RNA. So we're going to see more detail, but that is the majority of it. Some people, when talking about the central dogma, also look at how DNA is copied, because when a cell divides and tries to become two cells, it needs to first copy all of its DNA. And so that often is part of it. And the DNA molecule is copied by an enzyme complex called DNA polymerase. So RNA is made by RNA polymerase, DNA is made by DNA polymerase. Okay, polymerase.
So I've said the word polymerase or polymerase several times, and I want to unpack that a little bit. So polymerase has two parts, polymer and ace. So why don't you pause this video and ask yourself, what does ace on the end of a word mean? Hopefully what you thought of is an enzyme. The names of enzymes typically have ace at the end. And then polymer. And a polymer is a big molecule made of smaller repeating molecules. So let me tell you about enzymes and let me tell you about polymers. So one polymer that you might be aware of is, this isn't going to work well, but polyvinyl chloride, or PVC. We make pipes out of it. This is a giant molecule made by connecting vinyl chloride molecules together. Vinyl chloride is a tiny molecule. It is very small. It's smaller than glucose. So how do you make a pipe out of a molecule that is smaller than glucose? But that really is what it's made of. They react all those vinyl chlorides together to make a giant molecule, and realistically the pipe isn't one giant molecule, but there are giant molecules in it made of just connected vinyl chloride. Okay, so that's a polymer. And DNA, RNA, and proteins are also polymers, but instead of vinyl chloride, they're made of either nucleotides for DNA and RNA, or proteins for, or amino acids for proteins. So while we're here, let's define enzyme. An enzyme is a protein that catalyzes, I think that's a Z. It catalyzes a chemical reaction. Okay, so why do we care about proteins that catalyze chemical reactions in cells? It's like this. Cells have lots of different molecules inside them. Lots and lots and lots of different molecules. And those molecules can react with each other, but generally they don't. And so if a cell decides, I want this reaction to happen, all it has to do is make the enzyme that catalyzes that reaction. And then that reaction will happen. So it can turn chemical reactions on and off by making an enzyme that catalyzes them, and then waiting about an hour for that enzyme to break down. And then there's no catalyst and that reaction stops. So cells have a lot of other ways of modifying enzymes once they make them, but enzymes really are critical to everything about a cell. So there is that. Let's go back to the slides. Okay, here we were. Each protein has, a distinct function. An enzyme is one of those functions. A human cell can make something like 20,000 different proteins, many of which are enzymes, and a bacterium can make 1/10 of that usually. Many of them are enzymes. What else can proteins be? Well, enzymes are my favorite. I like to talk about enzymes. I want you to think about enzymes. But a lot of them are also structural. You could think of actin and myosin, and those things make... Myosin's a bad example. Tubulin that makes microtubules is a better example. Actin is the best example. Structural. They're just, you build things out of proteins. And then regulators help a cell decide to do things. So they're basically enzymes that are going to make other enzymes work differently. And that's pretty cool. So these are functions you should know. These are possible things. Because when we think about the central dogma and the cell deciding to make a protein or not, why is it doing that? Why is a cell always in the business of making proteins? Partly, it's to control its internal chemistry by making enzymes. Maybe it needs to do these other things. So there's that. And then let's probably stop this video here. So this is probably enough for this video. I just want to leave you with this thought on this very colorful slide. And this is something you should burn into your brain when you think about cells. And this is a class about cells. Synthesizing the correct protein at the correct time, in the correct amount, is every cell's full-time job. Every cell in your body, every bacterial cell, every fungal cell, all of them, their full-time job is making the right protein at the right time and the right amount. So with that, I will see you at the next video.